All right, histology is starting with the periods of prenatal development. Histology in general is the study of microscopic structure and function of tissues. And we are going to talk about the periods of prenatal development and kind of what happens within those periods in this PowerPoint. This chart shows the 3 periods of prenatal development. The first column shows the pre-implantation period, which is occurring during the first week. We have the pre-implantation implantation formation of embryonic disc, starting with the zygote, ovum and sperm meeting, cleavage. The zygote undergoes meiosis and splits into more cells. Then we have a morella on the 4th day, and then the blastocyst implants on day 6. During that second column, the 2nd through the 8th week, is the embryonic period. Different types of tissues are developing and organizing. During this embryonic time period, birth defects are the most common. And a lot of the birth defects or congenital malformations or even miscarriages occur during this time period, the embryonic period. Then from the 3rd to the 9th month, so the last 2 trimesters, is the fetal time period. As I said, most birth defects occur during that pre-implantation period and the embryonic period during the first trimester. So really the first week it doesn't implant, something happens there, or the 2nd through 8th week. Now, the congenital malformations can be caused by things that are genetic, environmental, teratogenic infections, drugs, radiations. Some of the examples that I gave in class were rubella or measles can result in deafness, cataracts, or cardiac defects. And syphilis, which is caused by the spirochete treponema pallidum, produces defects in the incisors and molars. So we've got Hutchinson's incisors or mulberry molars that can occur because of a teratogen, which is syphilis, that can affect the developing fetus. Any congenital malformations can be traced to specific time periods in embryological development. The pre-implantation occurs in the first week, and we've got the zygote, which is the ovum and sperm. Then we've got cleavage that occurs. The zygote undergoes meiosis and splits into more cells, and it becomes a morella, which is a ball of cells, on the 4th day. And then the blastocyst implants on day 6. The blastocyst is composed of a trophoblast, which is an outer cell layer, and an embryoblast, which is a group of cells across the middle, starting specifically with the bilaminar disc. So there's 2 layers that are going across that, which we'll see in the next slide, a picture. We're going to look at the diagram on the next slide, and we're going to look at where the zygote is compared to the blastocyst. And the blastocyst cells will be pushed to one side, and then the morella is when it's just a ball of cells. So between the zygote stage and the blastocyst stage, which occurs about day 6, right before implantation on day 6. And when we look at this, it's amazing to me how quick everything happens. So we've got the zygote, the ovum and sperm, then we've got cleavage that occurs, so the zygote undergoes meiosis and splits into more cells. So there's 2 cells in 30 hours, there's 4 cells in 40 to 50 hours, there's 8 cells in 60 hours, there's 12 to 16 in 4 days. We've got the blastocyst, where we've got that morella ball of cells that is kind of over to the one side, and implantation occurs on day 6. So it's implanted in the side of the uterus, and then we've got this outer trophoblast and then that inner blastocyst area. Implantation occurs on day 6, and the end of the embryo that's implanted in the uterus is called the embryoblast. The circle of cells surrounding the embryo is called the trophoblast. So we've got the outer trophoblast, which are the cells around the embryoblast. We have the trophoblast itself that gives rise to important prenatal support tissue, so a layer right under that outer trophoblast. And then we've got the embryoblast that gives rise to the embryo during the prenatal period. The end of that is implanted in the uterus on day 6. During the embryonic period, which is the 2nd through 8th week, birth defects are more common. During this time period, we have specifically induction, proliferation, differentiation, morphogenesis that happen before the full attainment of adult size and function within the maturation stage. You have to know these in order. You have to know that induction happens first, where the action of one group of cells on another leads to establishing a developmental pathway. Then you have to know about proliferation, where cells grow and divide, differentiation, where they become specialized and know what their job's going to be, morphogenesis, they're changing into the shape and structure that they're going to mature into during the maturation time period. Induction is the action of one group of cells on another that leads to establishing developmental pathways in responding tissue. We'll hear about induction in a lot of different histology growth pathways. Proliferation, cells grow and divide. There are two different kinds of growth that we have during proliferation. One is appositional growth, where the tissue enlarges by addition of layers on the outside of a structure. So when we're looking at hard tissue growth, so bone or hard dental tissues like enamel, there's appositional growth. So there's layers upon layers that get added to that structure. There's also interstitial growth, which occurs deep within tissue or an organ. This is the way that soft tissue, skin, or gingival tissue cells grow and divide, through interstitial growth occurring deep within the tissue or organ. In differentiation, cells become specialized and know what their job will be. There's cytodifferentiation, which there's development of different cell types, and then there's histodifferentiation, where there's development of different histological tissue types within a structure. So you've got ectoderm, endoderm, mesoderm that we'll be talking about shortly. Morphogenesis is the process of developing specific shape and structure. The cells are starting to change shape and look a little bit more like their eventual shape. And then maturation is the attainment of adult size and function due to proliferation, differentiation, and morphogenesis. So on day 6, we have the blastocyst implants with the embryoblast on one side. During that 2nd through the 8th week is when there's going to be an increased number of cells, and there's going to be some cell layers that become evident. At this point, it consists of the embryoblast and trophoblast layer, like I described before. The embryoblast, the ball of cells, and then the trophoblast, the outside area, with the embryoblast being closest to the uterine wall. In this cross section, you can see the embryoblast layer and the trophoblast layer, and then the endometrium of the uterine wall where it's implanted. In the next slide, we're going to see that blastocysts start to change, and we're going to see a bilaminar disc that forms across. And I'll explain the superior component of the epiblast and the inferior component of the hypoblast in the next slide, because that bilaminar disc is going to develop into the actual embryo. So in this picture, it shows the bilaminar embryonic disc that is starting to form. We've got the placenta on the side. We have the top layer, or the superior layer, which is the epiblast layer, which will eventually become ectoderm. Then we have the hypoblast layer, or the inferior layer, which will eventually become endoderm. At this point, it's the epiblast and the hypoblast layer, and it's 2 layers, so it's a bilaminar disc. We've got the amniotic cavity of fluid that is above that, superior to that, and then the yolk sac that's below that. The yolk sac is going to be initial nutrition for the disc, and then we have the 2 layers across there at this point, so a bilaminar disc. During this 3rd week, the primitive streak is present across that. So that is the bilaminar disc. It kind of differentiates into the primitive streak, which is a rod-shaped thickening in the middle of the disc on the epiblast layer. The cells from the epiblast layer at the primitive streak migrate towards the hypoblast and become mesenchyme. So I like this bottom right picture where it shows the epiblast and the hypoblast layer, which are the bilaminar disc. We've got the primitive streak in the middle. We've got the amniotic cavity above, the yolk sac below. The epiblast moves towards the hypoblast, and the epiblast is going to eventually become ectoderm. The hypoblast is going to eventually become endoderm, but the epiblast layer moves towards the hypoblast area in the primitive streak. The cells between the epiblast and hypoblast are going to eventually become mesoderm from mesenchyme and neural crest cells kind of mixing in there. There's a lot more to it than that, but for our purposes, that's kind of where we're gonna, as deep as we're gonna go. A little bit more about mesenchyme. Mesenchyme is made of mesenchymal cells, which is embryonic connective tissue. It can develop into lots of connective tissue and lots of different things. Some mesenchyme, when mixed with neural crest cells, is going to form that third layer called mesoderm. The action of the epiblast moving towards the hypoblast and the induction of the mesenchyme and neural crest cells ends up giving you another germ layer. So we would have ectoderm that came from the epiblast. We would have endoderm on the inside that came from the hypoblast, and the induction of the epiblast on the hypoblast with the mesenchyme and neural crest cells will give us mesoderm, the middle layer. Once we have those three layers that are now present in the embryo, it is going to be a trilaminar disc. The epiblast giving rise to ectoderm, the hypoblast giving rise to endoderm, the mesenchyme that was created from the induction of the epiblast touching the hypoblast and adding in some neural crest cells there will give rise to mesoderm. Ectoderm on the outside, mesoderm in the middle, and endoderm on the inside are all called germ layers, tooth germ layers, and all other tissues originate from ectoderm, mesoderm, and endoderm. Now we have the epiblast layer, the superior layer with high columnar cells, and then we have the hypoblast layer, the inferior layer with small cuboidal cells that migrate towards each other, the epiblast specifically into the hypoblast. So we've got some migratory cells in this picture that are working on the primitive streak, and those are all going to develop into the three separate germ layers: the ectoderm from the epiblast, the mesoderm from the migratory cells and the mesenchyme and the neural crest cells, and then the endoderm coming from the hypoblast layer. And it's going to go from an embryonic bilaminar disc to a trilaminar disc with three layers. This slide shows a table of the embryonic cell layers. It shows the ectoderm originating from the epiblast layer and those high columnar cells. The future systemic tissues include the nervous system, sensory epithelium of the eye, ear, nose, epidermis, hair, nails, mammary, cutaneous, salivary glands, skin, epidermis. Specifically for our purposes, we need to know that tooth enamel is derived from ectoderm. Mesoderm originates from the migrating cells from the epiblast layer going towards the hypoblast, and then we've got the mesenchyme and the neural crest cells in there. The morphology of the structure varies depending on which type of tissue it is, and remember this is the middle tissue, so this would include muscles, connective tissue derivatives, bone, cartilage. Specifically for us, we need to know that dentin, pulp, cementum, and PDL originate from mesoderm. And then we've got endoderm, which is our inside layer that originates from the hypoblast layer, small cuboidal cells, And the endoderm is the GI tract and associated glands and the respiratory epithelium, and then also the liver and pancreatic cells. Now on the end, we've got neural crest cells listed as a embryonic layer, and histologists consider neural crest cells kind of like a fourth embryonic cell layer. The neural crest cells develop from neuroectoderm, and the neural crest cells come from the crest of neural folds. Neural crest cells plus mesoderm equals mesenchyme, and mesenchyme in particular is involved in the development of many face and neck structures such as the branchial arches. So there's a lot more underneath the surface for neural crest cells, mesenchyme, and ectomesenchyme, which is specifically a specialized form of mesenchyme for formation of the pulp, the dentin, the cementum, alveolar process, and some of our tooth structures there. So this is our table that shows the development of those embryonic layers we've been talking about. Now our trilaminar disc now has a head end or cephalic end, and then it has a tail end or a caudal end. The cephalic end is going to be where the future site of the oropharyngeal membrane is, so the back of the throat or the back of the mouth. And then the caudal end is the future site of the cloacal membrane, which is where the anus or rectum area would be. The previous slides alluded to the fact that this trilaminar disc kind of looks like a flip-flop, and after the formation of mesoderm, when there are 3 layers, so it went from epiblast to ectoderm, hypoblast to endoderm, and then the mesoderm was formed by the migrating cells and the mesenchyme and the neural crest cells. So we've got 3 layers, trilaminar disc. It starts to take on a little bit of a flip-flop shape where we've got a cephalic end or a head end and then a caudal end or a tail end. The oropharyngeal membrane is near the cephalic end, and the cloacal membrane, that would be to the anus on the other end, is on the caudal end. Central nervous system development begins at the end of the 3rd week, and then specialized cells from ectoderm give rise to neuroectoderm. We're going to see in the next slide that when the CNS, or the central nervous system, is forming, a neural groove forms from the deepening of the primitive streak. The neural groove deepens and is surrounded by neural folds. And if you remember, neural crest cells come from the crest of neural folds. The neural tube is going to fuse to give us the spinal cord, and we're going to take a look at that in the next diagram on the next slide. Here the primitive streak is deepening, and the neural groove is folding and is going to fuse to form the neural tube in that bottom picture. As I mentioned before, neural crest cells, which histologists consider the 4th embryonic cell layer, they migrate from the crest of neural folds. They migrate through mesenchyme and are required for the formation of most oral and dental tissues, except for enamel and certain types of cementum. So for normal formation of the face, neck, and oral tissues, we need neural crest cells. Again, neural crest cells are vital to the normal development of the head, neck, and teeth, except for enamel and certain types of cementum, and some histologists believe that this should be considered a 4th germ layer. That's why there was a star on that table that we saw before. So in class, we talked about the different types of tissue. So the basic types of tissue, and we talked about epithelial tissue, the epidermis coming from ectoderm, and the dermis coming from mesoderm, and there being somites that arise from underlying mesoderm that originates in somites, which are masses of mesoderm on each side of the neural tube. When we look at connective tissue, and that's fibrous, loose areolar, adipose, hemopoietic, bone, and cartilage, all those middle layers, they derive from mesoderm, which came from the somites, which were clumps of mesoderm that were on each side of the neural tube that will become connective tissue structures. So there are different types of somites. There are sclerotomas, there's myotomas that would be responsible for future muscle development. There's dermatoma, which would be responsible for dermis, subcutaneous tissue, and visceral mesoderma. And all of those kind of are under the heading of connective tissue, but they come from somites, 38 pairs of somites. So mesoderm differentiates into paired clumps of cells on each side of the developing midline or future spine and gives rise to most of the skeletal structures of the head, neck, trunk, dermis, and associated muscles. We also have muscles under that mesoderm category, and there's smooth involuntary, striated voluntary, which are skeletal muscles, and striated involuntary, which are cardiac muscles that also fall under the somites, kind of leading to those different structures. And then our fourth basic tissue behind epithelial, connective, and muscle is nervous tissue, and that comes from ectoderm. Here's a picture of the developing nervous system with the somites on either side of that spinal area. And when we look at the side view, it's starting to look a little bit closer to what we would consider an embryo, you know, almost like a little, where it's almost got a head end and the cephalic end or a caudal end. And different mesoderm gives rise to the somites that are on each side of that nervous system. So during the fourth week, embryonic folding occurs. We've got the endoderm that's now inside the ectoderm. Remember, the endoderm is going to be the lining to the respiratory epithelium and the digestive tract, and then the ectoderm is going to be the outside type things like the skin, the epidermis of the skin, the tooth enamel, and the nervous system. And then mesoderm is going to be the muscle areas and the connective tissue areas, which are in between the inside and the outside of the eventual embryo. Here it shows the folding of the embryo. The trilaminar disc has folded into the embryo as a result of the growth of the ectoderm. The ectoderm's on the outside, the endoderm's on the inside, and then the mesoderm is starting to fill in the in-between areas. The 9th week to the 9th month starts the fetal period. So during this time, there's going to be maturation of existing tissue. Newly formed tissue is going to begin to function. We're going to go into the palatal formation, the formation of the face, the formation of the tongue and the teeth. And it's amazing to think that tetracycline staining of a child's teeth when a mother takes tetracycline would occur during this time period in utero.